Hello, and a very warm welcome to all to today's ISNTD Connect. I hope you can hear me well because there's been a few internet glitches in the run-up to this event, to this live session, but I think we're good to go now. If you're new to this series, well, welcome. We meet as regularly as possible just to have an opportunity to learn from each other, learning and sharing from colleagues around the world. And it's really exciting that today, under that umbrella and against that background, we are going to launch what we call Taking a Walk on the Lab Side. And with immense pleasure, just kind of getting to know about the colleagues around the world working on research in neglected tropical diseases, perhaps finding out a little bit more about how their labs are set up, what brought them to their discipline and to the projects they're currently working on, and just sharing best practices, perhaps answering some of your questions here in the audience. So it's my pleasure to welcome you all today. Pleasure to have our two guests online as well. And so without any further delay, I would like to welcome very warmly here up on screen with us today, Dr. Edward D'Antonio. Welcome.
Hi, thank you for having me.
Hi, you are Professor of Biochemistry and Structural Biology in the Department of Natural Sciences at the University of South Carolina Beaufort in the USA. I won't say any more about what you do because that's part of the session. We're going to discover that. And also joining us today, Dr. Miguel Chávez Fumagalli. It's a pleasure to have you here today, Miguel.
Thanks for having me.
Welcome. And you're head of the Computational Biology and Chemistry Research Group at the Universidad Católica de Santa María in Peru. A very warm welcome to you both.
Thanks.
Our online audience is joining us. If you have a moment, please don't hesitate to jump in the chat and tell us who you are, where you're connecting from. It's always lovely to hear from you. So please do that. Any questions, any comments, and anything you would like to share with our speakers and just the group in general, please go ahead, add that to the chat. But first and foremost, let's take a walk on the lab side and find out who's here today with us on the call. We've got Miguel, you're joining us from Peru. Edward, you're tuning in from the USA. Perhaps I'll hand over first to you, Miguel, to tell us a little bit more about what you do. We have a very diverse audience, very cross-disciplinary, very international. So please, over to you to introduce your work.
Hi, thanks. We are working with computational biology and chemistry for developing new drugs and vaccines and new candidates for diagnosis for the neglected tropical diseases. My main specialty is leishmaniasis, but we are trying to work with Chagas disease, Hades, and that's what we are working right now. The main specialty right now is trying to develop drug candidates for visceral leishmaniasis, and that's what we are doing now.
Wonderful, thank you, Edward. You're also leading some quite groundbreaking research in new treatments for neglected tropical diseases. What is your group working on?
My group works on primarily Trypanosoma cruzi, the etiological agent for Chagas disease. We study a potential drug target found in that organism called Trypanosoma cruzi glucokinase. And we're also venturing to look into the hexokinase. That is, these two glucose kinases are responsible in creating G6P. It's a metabolite important for 3 metabolic pathways. And essentially what we're trying to do is we want to inhibit these two enzymes. because we think that they're the most, they're important drug targets for the organism. I also study Trypanosoma brucei, the etiological agent for sleeping sickness, and I look at leishmaniasis as well. We have a project on arginase, and those are the main pathogens that I study in terms of the neglected tropical diseases realm. We also are investigating Toxoplasma gondii, and but that's more of a just one of these neglected diseases. So maybe not so much tropical.
Thank you. And Edward, have you always, has this always been the focus of your work, or what was your journey to this point in your career?
I was a graduate student back in 2004 in the Department of Chemistry at North Carolina State University, where I got into graduate school just being a chemist. And I went through the years learning biochemistry just at the basic level. And when it was 2010, I knew that I would like to go and become a postdoc, and I went to the University of Pennsylvania. And at that moment, I could study something where it was biochemistry and medically related, because prior to my postdoctoral fellowship, it was just general biochemistry. So I'd have to say that when I arrived at UPenn in Philadelphia, I began studying basically leishmaniasis at that point.
I think Maria has left the room. She had some problems with the
internet connection.
With the internet connection, yeah.
We could wait until she comes back.
Sure.
Although maybe Miguel, would you like to tell how you got involved into neglected tropical diseases?
I will take her issue and try to develop a same as you went. Well, I started at my master's degree with leishmaniasis, which is I was at the Federal University of Minas Gerais in Brazil, which is the one of the best universities in Brazil. And visceral leishmaniasis is a huge problem in Brazil. So there are many groups that work with this disease in Minas Gerais and other states. And I get involved in vaccine development. I started my journey trying to develop a vaccine that would help to control the disease in dogs, because I'm a vet. So I get involved in my master's degree, then in my doctoral thesis, and when I went to the postdoc, I developed some bioinformatics skills. So that's my journey started then with computational biology, and then I took the way to develop our work on drug discovery, and we think with main interest in natural products. So that's my my start was with vaccines. I went in many works with diagnostics, and my journey has taken me to drug development, and mainly with natural products. So it's a very diverse type of approach that I took for my research. Hi, Maria.
I'm alive. Sorry. Well, we're coming up to like 100 connects, and we've never had a major catastrophe. So sorry about that. But as our audience knows, this is completely live, and it's part of the process, I'm afraid. So I do apologize. I missed a few minutes, but we know each other well, because we've had the pleasure of hosting both of you on many of our ISNTD drug discovery meetings and conferences. So yeah, it's really great to get to hear a little bit more about your journey. So thank you for that. Why don't we just go and have a closer look at your labs now? I think you've set the bar very high, both of you, with this Take a Walk on the Lab Site series. We just meant to have just a short tour, but you've both created these amazing videos for us. So honestly, a huge thank you from us all at ISNTD. And without any further delay, let's fly over to Peru, to your university, Miguel. Let's find out all about your lab at the Universidad Católica de Santa Maria and the computational biology work that you're doing there. So over to the labs.
Sí.
Hi, this is Dr. Miguel Chávez Fumagalli. As head of the Computational Biology and Chemistry Research Group at the Católica University of Santa Maria, I would like to introduce the work of my research group is developing on the neglected tropical diseases. The group is working on natural product-based drug discovery against the neglected tropical diseases and is focused on visceral leishmaniasis, but not limited to, since we have ongoing projects on Chagas disease, rabies, and among other diseases. We apply computer-aided drug and target discovery methods for this endeavor. A combination of computational biology, cheminformatics, molecular modeling, molecular docking, and molecular dynamic simulation has allowed us to discover or finding new molecular targets and compounds from natural sources as potential drug candidates against the disease. Applying computational chemistry analysis has allowed the understanding of processes by which Leishmania recognizes, adapts to, and utilizes fluctuations in pH to unravel important features of its life cycle. This has driven us to discover prospective new molecular targets that are essential for the survival of the parasite inside the macrophage. Also, a combination of network pharmacology and single-cell RNA sequencing analysis is providing us with new insights into the potentially toxic effect on the human body of the compounds we are studying. Also, it's good to mention that the last year we launched the Peruvian Natural Products Database Initiative, which was envisioned as a springboard for biotechnological products and for providing suggestions for conservation policies.
The Peruvian Natural Products Database, Peru MPDB, developed by our research team, is a curated open access database that compiles metabolites from Peruvian plants and animals. These compounds have been carefully selected and compiled to provide research with reliable and comprehensive tools for exploring the therapeutic potential of Peruvian natural products. We are aware that neglected disease affects millions of people worldwide, especially in regions where resources are scarce and access to healthcare is limited. The richness of Peruvian biodiversity, along with the long history of traditional use of medicinal plants in the region, offer a treasure trove of natural compounds that could potentially contain treatment for this disease.
Okay, in the fight against leishmaniasis, we undergo different steps, one of which is virtual screening and molecular docking. This method helps us sift through vast libraries of ligands that might have potential inhibitory effect of target proteins. In our study, we docked over 3,000 natural products from 3 Latin American libraries, finding potential candidates with an inhibitory effect on mannose oligosaccharide glucosidases, an enzyme that plays a crucial role in the glycosylation process and correct development of leishmania. Through these techniques, we are able to predict at an atomic level the binding affinity of these natural products with the enzyme active center, and even reveal the nature and strength of these interactions. Of course, We understand that this approach will require further validation through biological assays. Scan the following QR code for more information.
So, in this opportunity, computational simulation technologies facilitate the resolution of intricate biomedical problems, enabling researchers to anticipate the behavior of natural systems under external conditions. We believe that it's important to analyze the behavior of proteins in different environmental conditions, such as solvent, temperature, and pH. Neglected diseases, such as leishmaniasis, analyzed by all-atom molecular dynamic simulations, allow us to study in detail the molecular interactions of different therapeutic targets. In this presentation, the genus of Leishmania infantum is depicted. This protein is located in the center of a cubic box with the presence of explicit solvent and the vary of temperature. Additionally, the presence of ions, of these ions, metal ions, is important to show in this work. For this reason, it's important to know the real behavior of macromolecules and thereby reproduce them using computational simulations methods.
As we saw, the scientists and students from the Computational Biology and Chemistry Research Group are working on the search for natural products-based compounds for the treatment of visceral leishmaniasis and other neglected tropical diseases. By applying modern in silico analysis, new insights into parasite biology have rendered to the group the discovery of new molecular targets with predicted importance on the parasite survival, while the potential of natural products' polypharmacology properties has been studied targeting more than one molecular target in essential pathways of the parasite. An important part of the work developed by the research group is on the curation and maintenance of the Peruvian natural products database. Peru is considered one of the 17 megadiverse countries, and the Peruvian population uses approximately 5,000 Peruvian plants for 49 purposes or applications, where about 1,400 species are described as medicinal. However, the number of species studied is less than optimal. Information regarding Peruvian fungi, propolis, bacteria, and marine organisms, among others, is absent in the literature. We believe that in long-term approach, natural product research may provide new drugs for the treatment of neglected tropical diseases, with higher effectiveness and less undesirable side effects of current medications. Also, by applying in the early stage of development, computational biology and chemistry analysis, the development of new drugs can be fastened while cutting costs in the process. All right, thank you.
Fantastic.
Thanks.
Yeah, and thank you, and a huge thank you to all your collaborators. Your amazing research, very exciting, and also this growing Peruvian natural products database initiative. Some really exciting work there, and there's been a question in the chat for you already, Miguel, from Yasmin Razi. Welcome, Yasmin, asking, good afternoon. Could you please tell us which software you use for docking?
Oh, it is. We don't have a software. We use I don't know, for virtual screening is Autodoc, and we use also Swiss docking or Doctor. It's not we don't have any specific one. We can use Chimera, so it's not as we only like one. We use as we. As we used to.
Thank you. That's good to know. If there are any more questions for Miguel and the team, please don't hesitate to keep adding them in the chat. But now we're going to take another journey and go to University of South Carolina Beaufort. Edward, we're ready to discover your lab. So we're going to play the second video. It's also about 10 minutes long. And then we'll come back and ask you lots of questions as well. Here we go.
Greetings, my name is Dr. Edward D'Antonio, and I'm here in the United States at the University of South Carolina Beaufort. For the Take a Walk on the Lab side for ISNTD Connect, my undergraduate researchers will talk about their biochemical techniques that they perform in my laboratory. So let's begin.
Hello, my name is Lindsay Baker, and I am a biology major and an inspiring PhD student in the field of microbiology. I am very passionate about research, and I plan to make advancements throughout my career in medicine and research to better the lives of people who are suffering from disease. I perform research related to Trypanosoma cruzi, in which I study the enzyme glucokinase. Glucokinase is considered a potential drug target, but my research is involved in determining if it is actually a validated drug target. This project involves protein overexpression in E. coli and purification. In this case, we are purifying a mutation of this protein. After expressing our desired protein in E. coli bacteria, we collect the E. coli cell pellet through centrifugation that usually has a mass of about 35 grams. We can begin purifying our protein from the E. coli by lysing those cells. Our first chromatography procedure is an affinity chromatography using a column packed with cobalt NTA resin. Lysed cells are centrifuged for 45 minutes at a speed of 11,400 RPMs to collect the supernatant. The supernatant is then collected in 50 ml tubes and set in ice. We condition our column with buffer using a peristaltic pump. Impure supernatant immediately runs off the column, and our protein of interest will be collected after we change our mobile phase composition. After this process is complete, the column is connected to our FPLC instrument to remove any additional impurities and to contain the protein into glass tubes as fractions. We can see if the indicated peaks on the chromatogram reveals our protein eluting off the cobalt NTA column. The major peak is what we are interested in and when we change the composition of the mobile phase during the run. After further purification, we can use these proteins in enzyme inhibitor assays to determine their activity against inhibitory compounds. Now let's hear from my fellow lab mate, Anna.
I'm Anna Husett, and I work mostly with agarose gels and SDS Page in the D'Antonio laboratory. I am also currently working with the specific enzymes of Trypanosoma cruzi and Leishmania infantum. I also work with molecular taxonomy work. In molecular taxonomic approaches, I currently perform PCR and DNA extraction from agarose gels to then send off DNA samples to Sanger sequencing. I use specifically designed primers that bind to genomic DNA in which we observed an amplicon in gel electrophoresis. This type of technique becomes important in NTD research since I can use it to identify if an insect has the parasite Trypanosoma cruzi, the etiological agent for Chagas disease. My procedure involves running a PCR followed by running a typically 1.2% agarose gel with ethidium bromide and then observing the PCR DNA sample under UV light. Amplicon bands are cut out with a sterile blade and then a DNA extraction kit is used to isolate the purified amplicon DNA. And then the samples are sent off for Sanger sequencing that I previously mentioned. The nucleotide results we receive can then be matched from the GenBank database via a nucleotide blast search, also called an NBLAST, which are then compared to all sequences that have been submitted to GenBank, a site operated by the National Institutes of Health. With the sequence, we can then see what species it belongs to, but this is just part of what I do. I also work with enzymes. In this case, I perform overexpression and purifications. I grow E. coli bacteria, lyse cells, and isolate the protein of interest by rounds of chromatography. This process is known as fast protein liquid chromatography, FPLC, in which I use a high-pressure pump system in combination with a UV detector and fraction collector. We use different columns that are attached to the pump. After I've purified an enzyme, I have to determine that the sample I have is actually pure. The best way to determine the purity of a protein is to run an SDS page, which is sodium dodecyl sulfate polyacrylamide gel electrophoresis. Once a gel has been stained with Kumasi brilliant blue dye and then destained, I can then observe if my protein sample is pure. From this step, you can also perform a trypsin digestion LC mass spec, where I would need to cut out my protein sample from the gel, expose my band cutouts to the enzyme trypsin, a protease, and recover the fragments. If you have a pure protein, then you can use it for assays to test for Michel's Menten kinetic parameters and for inhibitory activity, much like you will see with my friend and colleague Carson.
I am Carson Fry, and I'm currently working on a research project that involves early-stage therapeutic drug discovery for particular neglected tropical diseases, or what we call NTDs. The type of assay that I personally work with is wild-type Trypanosoma cruzi glucokinase. It is a potential drug target. I am testing derivatives of the cotton plant to determine if it has the inhibitory effect to the enzyme we work with, but is also against T. cruzi parasites. I run assays in the lab to see if they are good inhibitors. To initiate an assay reaction, we begin by adding ATP and terminate our reaction with EDTA after the optimal time has been reached. In my particular assay, the specific time is 120 seconds. Once the assay is meticulously separated into the different individual wells of the CoStar plate per the specific assay compounds in the correct order and microliters, they are then transferred into a new CoStar plate of 70 microliters each, which I then transfer with a multi-channel micropipette. I then take the assay plate to the fluorescent microplate reader to begin a plate scan. I personally work with Dixon plots, but others may work with Michaelis-Menten plots, which help extract biochemical parameters from the collected data. In the method of Dixon plot analysis, I am processing the data in order to determine the inhibitory constant value, also known as a Ki value. This value indicates how strong the compound inhibits the enzyme. And now you'll hear from my lab partner, Destiny.
My name is Destiny O'Neill, and I'm a pre-med undergraduate student. In Professor D'Antonio's research laboratory, I work with Carson Fry on therapeutic drug discovery projects. Carson and I use enzyme inhibition kinetics-based experiments to evaluate the inhibitory potential of natural products such as gospel, along with its synthetic derivatives against the potential drug target Trypanosoma cruzi glucokinase. To run the enzyme inhibition kinetics experiments, we manually perform assays, often running 2 compounds at a time, using a series of reagents and carefully calculated measurements including varying substrate concentrations. For each compound run against TC-Gluc, we use the Cornish-Bowden Dixon plot analysis method through Excel to generate processed data plots that provide insightful information on the mode of inhibition in addition to the respective inhibitory constants, or KIs, of each compound. The D'Antonio laboratory is also a collaborative laboratory, so Carson and I will be working in collaboration with NYU Langone Health's Anti-Infective Screening Corps, directed by Dr. Ana Rodriguez for in vitro toxicity screening of our discovered inhibitory compounds. I'm also excited to continue working on another project that I have taken on involving antibiotic property research on bacteria collected from a great white shark, where I have the opportunity to collaborate with not only USCB's own Dr. Kimberly Ritchie and her students, but also with Dr. Gustavo Mercaldi, who's based in Campinas, Brazil, at the Brazilian Biosciences National Laboratory. Working on the antibiotic project requires microbiology, compound isolation, and the use of various compound characterization techniques. The isolation of the biologically active compound will then be tested against Trypanosoma cruzi and other parasites that cause NTDs. I'm excited to see if any of the discoveries from this project can help identify novel antiparasitic compounds.
In conclusion, you've seen my labs now and the students that work in my research group. And I wanted to express a thank you to the ISNTD director, Marianne Comparet. And if you have any questions, please reach out. Thank you.
Another fabulous video, and I think it's safe to say it's the first time there's been a shark on an ISNTD Connect. So that's quite something. Thank you so much for sharing. It's been fascinating, and I'm not lab-based, but I'm sure that really being able to see visually how things are laid out, how different departments are working together, or just your setup in general is really enriching for many of our colleagues around the world. So thank you so much, and beautiful videos, amazing. Like I said, you've set the bar very high. Thank you.
You're welcome.
A few questions though. One of the things perhaps that is quite striking, particularly in the US, the labs are really named after the PI, the principal investigator. And so perhaps a question first for you, Edward. What stands behind that in terms of what that means for the research? Because it's not something that's necessarily found in around the world.
Yeah, it's mainly like more of a professor-centric viewpoint, I guess you could look at, because I know that one of my friends in France, and he was explaining to me that how in his laboratory, it's sort of like based on their department. But when in my case, I make the decisions for the research that I would like to do. I write the grants that I want to write, and you're supported by your department here because they're my colleagues, but you kind of get this freedom to do what you'd like. But it's also like there's like the survival aspect to maintain and be an independent researcher. So, which in my case, you have to write grants to be able to fund your lab and buy the equipment and things like that. So there is that responsibility to that. Maybe an added pressure for some, maybe not for others. But in my case, I haven't had too much trouble with that, and things have been smooth sailing for the past 11 years.
That's really great to hear. And it's actually true. Sadly, sometimes you do see labs shutting down. And so a system with both positives and challenges, a lot of responsibility, as you were explaining. How does that contrast with your experience, Miguel, as principal investigator on many projects?
Well, our research group is quite new. We have about 2 years of functioning. We got gathered together 2 PhDs, which are research associates, and Myron, which is my PhD student, which is professor from the pharmacology department. And we get together and we put together the research group. Right now we are turning to a lab. Right now we begin with a research group and we are becoming a lab. One advantage is that all of them can write their own project. They have Professor Barazorda have their own projects, founding, and so I think it's advantage for us, and we can share the advisory of students, and we have became in 2 years a 15-person group. So it has grown faster than I was thinking, and it has turned out that we are becoming a leader group in the country. So in only 2 years is quite surprising for me, and it's a happy thing. So we don't have any issues with granting. Everybody has their own experience and expertise. So we got together and we got working, trying to build something together. That's the spirit of the group.
Wonderful. Well, it sounds like you've been kept busy enough, so perhaps my next question is not yet of the moment, but have you found that you are kind of encouraged or wanting to connect to other departments, other laboratories, or is that changing for labs around the world, particularly in neglected tropical diseases? Is there like a movement to build either international collaborations or cross-sector ones?
Well, as Peru has no strong formation of PhDs, most of the scientists that work here are they studied out of Peru. So when we come back, we got together with our forming labs. So I have plenty of collaboration with Brazil. Dr. Goizueta also, Dr. Barazorda, she became a doctor in University of Barcelona. So all together we have a very strong network. We are having a first attempt with Dr. De Antonio too. With Edward, we are having a first work that we are trying to put together, and so on. I think it's a movement. We need to work together. For example, my lab, it's only computational lab, as you may see. We don't have the equipment that Edward has. So, but we have workstations that can do plenty of analysis. So we need to have collaboration in order to have an in vitro assay or an in vivo assay validation of our compounds. So it's a necessity. It's part of the spirit of our group to build strong networks, and we are open to collaborate with everybody. Once we have a conversation first, we can see that something we can work together. We are happy to collaborate.
Wonderful. Edward, which way are your collaborations going? And I think the fact that you're even on this series and you've really engaged with the ISNTD a lot, and as Miguel mentioned, both of you really fostered and developed this collaboration between your labs after meeting online, I think, if I'm not wrong. Which way are your collaborations going? Coming from a very different perspective and different country and environment.
Right. Well, through the years, I started out just trying to get projects up and running. And then I came across your organization back in 2017, where Cameron invited me to the Chagas's Roundtable, which was in Washington, D.C. And from that point, we had a chance to meet some like the NIH program officers at that meeting, and just kind of like getting the word around as to who I am as a researcher, trying to, and your organization has helped expose who I am a lot, farther from what I've done, even just from my publications. And then after COVID-19, because COVID-19, I had a big dip in my research program. I'm sure a lot of people have as well. But we ramped our efforts back up, and I saw one of the ISNTD Connects by Miguel, and I was excited because that was an enzyme I worked on when I was a postdoc. It was Leishmania mexicana arginase back at UPenn, and I had to get in touch with him and say, hey, let's collaborate on something, and and let's put our minds together, because experimentalists and computational biochemists, for example, can work off of each other, although the strategy is a lot different. Because computational, you're going to be able to see something very quickly, and it probably is going to be true. but the experimentalist must test it. And so that's where we can work off of each other and create good science. I've also been able to work with Sean Eakins from Collaborations Pharmaceuticals, based in Raleigh, North Carolina. That was an interesting collaboration. I've known Sean for a while from the American Chemical Society meeting, but we recently reconnected again after COVID-19, and there's other collaborative efforts. I went to the DNDi meeting, the Lola 10 in Campinas, Brazil this past fall, and I reconnected with one of my collaborators, Dr. Artur Cordeiro at Laboratório Nacional de Biociencias there, and we kind of talked about looking at different ways that we could solve the problem in Chagas' disease. Could we create a small team and figure out, let's develop a project, and we had some fruitful discussions over a dinner, there in Cambuí. So it was nice to reconnect with Artur. Yeah, so I mean, people have been drawn to the work I've done, and I've been drawn to theirs as well. But I'm all about let's try to get a project going, up and running, and let's make some progress.
I think that's a great phrase, I'm all about, because ultimately it does come down to the leadership, the type of scientist that you are, and that's something that's evident from how you've presented your labs and your work. It's really this openness to collaboration and quite an interesting ecosystem of partners that you just shared with us now, Edward and Miguel. So thank you for that. So lots of success, lots of momentum in both your projects and your labs. Some of the challenges, what have been some of the hurdles you've encountered? Are they still there? What can be done about them? I'm not sure who wants to go first.
Edward.
I could go first. Yeah, I mean, there's always going to be challenges, but I like challenges. I like if we're not failing, then that means because I like to solve hard problems, and what we can do is one problem that I've been having trouble on was to express Trypanosoma cruzi hexokinase in E. coli bacteria. So I need to revisit this because I've changed my hypothesis in my latest publication in IJMS, where the glucose kinases work together. You have TC-glucan hexokinase, but we need to now study hexokinase to verify, because we're trying to figure out, are these glucokinases the two drug targets? Are they like a dual drug target scenario? But efforts are definitely going on to that, and we're going to solve that problem soon.
Wonderful.
Well, my turn. As I see, there are some orders of problems. You have national policy problems, you have international policy problems, but it can be taken. I don't know. I don't think that it can be suitable to talk about them because they are very different, and my country is have some problems with that different, and have some with other countries you can share problems as technical problems for computational analysis on working on neglected tropical diseases. We have several. When we saw a nice figure at Nature or Science, we got these amazing figures of computational analysis, and for studying the type of disease we are working on, we have several vacuum of information. Many of the databases of type of analysis of several that kind of databases, they are not specialized for the kind of disease of we are working. So it's not only neglected as founding or type. They are neglected in information, which is very hard to understand. You know, an example can be that almost 60% of the genome of this parasite, like Leishmania or Trypanosoma cruzi, we don't know how they work. Most of them are hypothetical proteins that we don't know how they do on the parasite. So to apply this kind of analysis, we have to go to a gray area and have some background to ensure some kind of approximation. So it's a hard work to do, but it's a necessary hard work to do, because it can be, as is well known, bioinformatics and this type of analysis can faster some processes and can cut costs and can bring you some new targets or some drugs that can be cheaper, or can be it can be easily synthesized, because when we have some compounds that are very good, but they are very costly to synthesize, or are very hard to synthesize, which make the potential drug to rise the costs. So these all things can be achieved by informatics or by in silico analysis, but there are very, very difficult to point out vacuum of information. So it's like we need to work and try to diminish this information, this lack of information.
Yep.
May I add to another problem that seems relative to what we're saying? One big problem is customs in like Southern American countries, because, for example, like if I'm going to send a compound of interest to a collaborator in Brazil, it takes a very long time to arrive to its destination, where, had I sent it to somewhere in France, or, I could overnight that, and it'll get there to the United Kingdom or to Europe much faster than it would, and we're talking months. And wow. It would be amazing, if there could somehow be a law where, if it's, you're doing therapeutic drug discovery research and you need to send a compound to a collaborator, that the customs, there should be something to the customs agents that they're yep, we can let this pass. They've got the right paperwork. I mean, unless I'm doing something completely wrong, I've been, maybe Miguel might know something about this, but I've always had this roadblock with sending compounds to collaborators in Latin America.
Yeah, the last time we sent from United States to Brazil a compound, we bought it in the United States and they ship it to Brazil. It takes 2 months. So if you have a unstable, or it has some kind of loss, you have lost 2 months and a bunch of money. So
so on that topic, usually when you send a compound for the non-chemist, you want to have that compound on dry ice where it's going to be ice cold. If you send it at room temperature or ambient temperature, it'll degrade. I mean, you have compounds that they're not stable, especially some of these natural products that Miguel has, they would degrade within a day, being at room temperature. So they have to be shipped on a very cold temperature, and it needs to be placed into a freezer immediately. That's the problem with collaborating maybe with someone who works in another country for me, is that I have to ensure the quality assurance of those compounds have the highest integrity. And what has been happening is I would send my compounds, they would get there late. We also have to get taxed. Like you have to pay like a double tax in a way. And then, so let's say like you can get through all of that. Then when the compound is received, you would have to then do an HPLC analysis, and it's just another bottlenecking of work that seems to be so unnecessary.
Yep, that's a kind of problem.
Wow. That's been really fascinating, and I thought that the field of tropical diseases was quite good at these cross-border collaborations, but it is a huge challenge. Really interesting to kind of see that from your perspective. Hopefully, the fact we have really strong private-public partnerships in this field, also the ongoing discussions around, although it's a slightly different topic, but around the pandemic preparedness talks, are really looking at data sharing, samples, and cross-border collaborations. So hopefully there'll be some improvement there. We are coming up towards the end of our session. We have a few minutes left. So if anyone has questions, please do pop them in the chat. We've really learned a lot. It's been a fascinating 60 minutes that we've had to spend together. But just to kind of leave on a high note, did you have what might be some of your fondest memories, or perhaps an anecdote, or just some advice that you could give to those listening in on the calls? Running a lab is not easy, particularly in this field of tropical diseases. Resources come and go, don't they? Anything that you'd like to share in these last few minutes as a concluding remark?
Would you like me to go first, Miguel?
Sure.
Well, in medicinal chemistry, every day is like, discovering something fun. So usually a chemist will tell you that what I'm working on right now is the most important or most fascinating thing that I'm working on. Although I do have fond memories of discoveries that I've had, not only of what I'm working on now, but also my lab members. We have a great team, and we try to encourage everyone to have a happy lab. I think that's one of the key things when operating a lab, is that you have this coherentness. You create a lab culture that is hardworking, but also everyone is nice to each other, and that helps facilitate productivity. So if you're like trying to be an assistant professor, let's suppose, and you're thinking about, hey, that I want to go into academia, I want to get into NTDs. That's why you'd be here. I would say you have to have a set of principles going in, and to create a hardworking lab culture, and try to solve some really hard problems. Like go to conferences, get out to conferences every year. Like I always go every spring. I go to 3 conferences. In the spring, I go to maybe 2 conferences in the fall at least, because I want to know what's current. I want to talk to people who are at these conferences. You want to get yourself known. But I could tell you a quick fond memory. Basically, the best discovery I had was when I worked in Brazil in 2016 over the summer. I did a high throughput screen where we screened 13,040 compounds against TC Gluc. That's my potential drug target. And my collaborator told me, he said, because we're getting clusters of a chemical scaffold that's inhibiting your compound or your enzyme, that's a key result, because that means that molecule, that's a strong set of evidence that compound is important. Now, prior to that high throughput screen, I was a structural biologist. I only looked at things that would bind in the active site. But then we had new compounds that we just discovered off of this high throughput screening platform. And that really opened the door to a lot more publications that I'm starting to come out with now. So that was a pivotal moment, was to work with a high throughput screening robotics lab. Mm-hmm.
Fantastic. How about yourself, Miguel?
Well, as an advice, if you want to work with computational biology, chemistry, it has an advantage and a disadvantage, which is the same, which it depends on you. I am a vet. I start my research work with vaccine development, and by myself, I have learned in 8 years span all this world of computational analysis. I am not former with this kind of skills. So I learn. If I can learn, well, everybody can learn. So it depends on you. You have a notebook, and you have information, and you can have it all. As a fun thing, my first paper, I wrote the last paragraph of my first paper of bioinformatics in the room of my just born son. So it's like, yeah, he was just born, and I was working on. I had to stay at home because I had to help. So that's a driver of my turn to bioinformatics. Stay more at home, but I got to publish and to work. That's my. I'm a scientist. That's my issue. So I became a computational guy because I had to, and I learned all by myself. So it's as advantage for sure. You only need a computer. It's all you need. A disadvantage is all about you. You have to learn and to have a don't sleep or sleep 3-4 hours, and that's the issue. You have to work, and it's by yourself.
Yeah,
it's all on you.
Well, that's both very realistic, but also really encouraging that, as you said, what you need is a pen and a notebook, and the rest is up to you. The science is easy and people are complicated, so but I can see that, these two labs are in amazing hands and with really wonderful leaders, and we've enjoyed so much getting to know you and your work and your groups a bit better. Michelle Taylor in the chat has written, really great to get to see inside the laboratories. Thank you. And I think that would be very much the feeling echoed across all our audience. You know, the fields you're working in, bioinformatics, computational chemistry, molecular modeling, they're very specific disciplines, but it's been really nice to get to learn from you as well, for those who are working in a wide range of fields and approaches and research projects in neglected tropical diseases. So really a massive thank you again. We're coming up to the end of our time for this week's ISNTD Connect. It's also been a lovely pleasure to have our audience. And I'm so sorry, I just saw on the Q&A tab a question, if we have 1 or 2 more minutes to answer this question by Hafizu Mohammed before we go, who was just asking about saying, thank you for the presentations. Absolutely. And I'm interested in NTD's research. Can you give us the links to your lab websites? So I'll just copy that over back to the main chat. Absolutely. If you want to find out more, here we go. And Miguel, if you have a chance as well to copy paste your lab into the chat, I'm sure that'll be very useful for everyone. You've made it clear that you're open to collaborations and exchanges, so please, by all means, reach out. And we look forward to hearing a lot more from where your research is going. So a very big thank you. To our audience, when I ejected myself from my own webinar, which was very unprofessional, unfortunately, I lost the chat, so I didn't see who was saying hello from where. But a very big thank you to the few I can see: Deborah Charles, Alice Guiza Tembe, thank you, Trifin Tinago, Michael Peretz, Marian Candia. Well, thank you so much for joining us this week. We'll be back next week on a slightly different topic, which will be looking at the role of gender in trachoma and trichiasis. And if anyone at all who's been on the call has been inspired to come and share their lab with us in a future episode of Take a Walk on the Lab Side, then please do get in touch with ISNTD. It'll be an absolute pleasure to have you here. But in the meantime, another final round of applause for our speakers. Thank you so much. It's been a really enriching session. We've enjoyed it a lot. And we look forward to keeping in touch and hopefully to seeing you very soon again on a future ISNTD Connect. Edward, Miguel, thank you.
Thank you.
Thank you, Marianne.
Yeah.
Have a great rest of your week, both of you.
Yeah, same to all. And let's definitely keep in touch.
Sure.
Mm-hmm.
Thank you very much. All the best, everyone. Bye.
Bye-bye.
Bye.
Bye.